Good morning, good afternoon, good evening. I would like to welcome you to the I4C 22 scientific session. My name is Philip Lupo. I am a professor of pediatrics at Baylor College of Medicine and the director of the Epidemiology and Population Sciences program at Texas Children's Cancer Center and also the chair of the Children's Oncology Group Epidemiology Committee. So I think you can see from our agenda, we have an exciting session for the day. And before we get started, I wanted to echo some of the previous speakers and note our disappointment for not being able to attend the meeting in person and enjoy the beautiful scenery and sights of Turin, which I hope will be an opportunity for us to join again and meet together in this beautiful location. With that said, I also want to thank the previous speakers for, I think, a phenomenal outline of the cohorts that are currently part of I4C. It's really exciting to hear not only about the individual efforts and the attempts for identifying cases in these cohorts, but also the amazing amounts of data and samples that are being collected to answer some really important questions. So without further ado, our first speaker is set, I believe, to take it away, and it's going to be Dr. Jeff He, who's going to be talking about maternal infection and the risk of childhood leukemia and highlighting recent findings from I4C.
Philip. Can you see my slides?
Yes.
Okay. Hello, everyone. I'm Jeff. I work with Terry in Oxford on the topic of maternal infection in pregnancy and childhood leukemia. And so one of the molecular characteristics of leukemia is fusion gene, which combines two separate genes incorrectly and result in abnormal gene expression and cell function. Previous studies showed that most of these leukemia-related fusion genes were present at birth. So this suggests that prenatal factors might play an important role in the development of childhood leukemia. And recently, several nested case control studies found that the leukemia cases had different level of cytokines at birth. So this suggests that immune-related factors during pregnancy may change the fetal immune system development and then increase the risk of childhood leukemia. Maternal infection is one of these suspected factors. Maternal infection could affect fetal health by different ways. So once the infectious agent reaches the fetus, it can lead to a lot of adverse consequences. For example, CMV, rubella, herpes simplex, and other pathogens are a classical group of teratogenic pathogens. And other bacteria can also lead to inflammatory responses, which in turn will increase the risk of fetal death, preterm birth, or fetal organ injuries. So how could maternal infection affect the risk of childhood leukemia? And this is my hypothesis. If the infectious agent is transferred from the mother to the fetus, and it could lead to genetic lesions or chromosomal modification in the fetus and then increase the risk of childhood leukemia. Another possibility is that the immune responses locked the infectious agent themselves could be transferred to the fetus and then change the fetal immune system development. So after birth, the child might have abnormal immune response to the childhood infection, which is a risk factor for childhood leukemia. Another pathway could be related to the perinatal outcomes. And maternal infection could increase the risk of birth defect, cesarean section, or preterm birth. And all of these adverse perinatal outcomes have been reported to be associated with childhood leukemia. So it is possible that a specific infection or infection in general is associated with the risk of childhood leukemia. And my research question is, in human being, is maternal infection during pregnancy associated with childhood leukemia? First, we did a systematic review and meta-analysis to summarize the evidence in previous studies. In this review, we identified 32 articles from 20 studies, including 15 studies on ALL and 14 on any childhood leukemia. And this is the main finding in our meta-analysis. So we identified 20 types of infections in previous study, which have been investigated for the association of any childhood leukemia. And we found influenza in the meta-analysis. Influenza was associated with higher risk of childhood leukemia. The pooled odds ratio is 1.77. And rubella and varicella were also associated with higher risk of leukemia. And the hazard ratio is high, but because of the number of studies was small, so the confidence interval of the effect size was wide. So we found influenza, varicella, rubella during pregnancy were associated with higher risk of childhood leukemia in previous literature. But most of these studies were based on case control design, and there were very few cohort studies in this field. And last, we used I4C data to reanalyze the association between maternal infection and childhood leukemia. At the moment, we have 7 cohorts in I4C, including one from the US, UK, Norway, Denmark, Italy, Israel, and Australia. But when I started my project, we only have data from 6 cohorts in I4C. So these 6 cohorts were included in my analysis. Totally, we have more than 300,000 mother or children in our database. And there were more than 500 childhood cancer cases, including 167 childhood leukemia. And this table shows how the maternal infection data were collected in different cohorts. And you can see that most cohorts use self-report questionnaires or interview to collect data. The third column shows the timing when the infection data were collected, and you can see that the timing of the data collection were a little bit different across the cohort. And this is the list of the infection variables included in my analysis. I only included infection variables which were collected at least in 2 cohorts and have prevalence of above 5% because of the number of cases in my analysis. And we can see that the respiratory tract infection had the highest prevalence, above 30%, and followed by common cold, influenza, vaginitis, and urinary tract infection. And we also have two variables on infection-related symptoms, such as diarrhea and fever. The primary outcome is any childhood leukemia. Secondary outcomes include ALL and AML. Most cohorts collect the cancer data through linkage to cancer registries. I use multi-level cost model to analyze the association between maternal infection and childhood leukemia. All models were adjusted for maternal age, educational level, parity, diabetes, BMI, and smoking during pregnancy. The missing data were imputed using multiple imputation. This is the result. I have three models in the analysis: an adjusted, semi-adjusted, and fully adjusted. Because the results were very similar between these models, so let's just focus on the fully model. So from this figure, we can see that urinary tract infection and its main time, cystitis, were associated with higher risk of childhood leukemia. The hazard ratio is 1.68. Influenza and any respiratory tract infection were also associated with higher risk of childhood leukemia. But the confidence interval of the effect estimate for influenza was wide. Other infections such as common cold, vaginitis, diarrhea, or fever was not associated with childhood leukemia. And then I stratified the analysis by leukemia subtypes. And we can see that the pattern of the association for ALL and AML were very similar to the one for any childhood leukemia I just showed in last slide. And for example, urinary tract infection and cystitis were also associated with higher risk of ALL and ALL. But the confidence interval of effect size was wide because of the reduced sample size in the stratified analysis. And again, respiratory tract infection was associated with higher risk of ALL and AML. And then I tried to investigate whether the infection timing could modify the association between urinary tract infection and childhood leukemia. And I found the association for UTI in late pregnancy appeared to be stronger compared to the one in early pregnancy. For example, the hazard ratio for UTI in late pregnancy was 1.9, but the one for UTI in early pregnancy is 1.25. But when I analyze the interaction effect, and this is not significant. I also performed several sensitivity analysis to test the robustness of the result. For example, after I adjusted for birth season or excluded baby born preterm birth or did a complete case analysis, the results were very similar to the primary analysis. So in using I4C data, we found a urinary tract infection and respiratory tract infection during pregnancy were associated with higher risk of childhood leukemia. This is the first large-scale prospective cohort data on this topic, but we relied on self-report data, so there might be a misclassification of the exposure. And another challenge is the heterogeneity in infection definitions, because different cohorts use different question or questionnaire to collect the infection exposure. Sometimes the question were a little bit different, or the timing was different. So the heterogeneity for the infection variable, for example, for influenza, sometimes was quite high. And another limitation is the moderate number of cases, although we have put 6 large cohort together. And as I mentioned, we only use self-report data, so we can only assess the infection in general or symptoms. So next step, I think it would be helpful to investigate the effect of specific pathogen. And another extension could be related to using different kind of data or related variables to analyze the association. For example, if we can access to the medical record data, this provides us an opportunity to assess the infection exposure in a different way. Another idea is we could use data on treatment or vaccination against the infection to analyze the effect. If maternal infection is associated the risk of childhood leukemia, treatment or vaccination against the infection might modify the risk. And another direction is to investigate the mechanism underlying the association we observed. Although we have several hypotheses, but the evidence for this potential mechanism is still not clear. I would like to thank my supervisors and colleagues in Oxford, and I also would like to thank my collaborators in I4C in Aarhus, Denmark, Norway, and I also thank my funding body, which support my PhD study in Oxford. Thank you.
Now at the University of Southern California, Dr. Joe Wiemels, who's going to be highlighting some of his really fascinating work on the epigenome and childhood leukemia. So, Joe, without further ado, I'll let you take it away.
Well, I'll just start talking while I get this set up. I'm going to talk about epigenetics and childhood leukemia today. And I'm going to talk about using epigenetics in 2 fashions. One is to understand the developmental origin of disease with the hypothesis that very early environmental exposures, especially those during pregnancy, can affect the future risk of cancer, and that those exposures are most likely to do that through epigenetic mechanisms. The other way that I'm going to talk about epigenetics today is how we can use epigenetic marks as biomarkers of exposures. So not in the fact that not exactly as those marks causing the disease, but that those marks can provide to us a window to past exposures. Okay. Try this again. It's telling me I need to quit WebEx to share.
Joe, I wonder if you'd like, you could send me the slides and I could advance them for you, or you know, just a thought to maybe facilitate. Unless you want to jump off and log back on.
I'm sending you the slides. Although I think it's going to be a big presentation.
Okay, we'll see what happens. We'll try our best.
Okay. Meanwhile, I'll keep talking.
Sure.
So as epidemiologists, the epigenetic mark that we have been looking at and are interested in is the most accessible one to us, and that is DNA methylation. So DNA methylation is a covalent modification of cytosines within DNA. So leukemia, like all cancers, is an epigenetic disease, and our studies of leukemia cells, the blast cells that form the disease, indicate to us or show to us that about 10% of the genome is altered in DNA methylation status. Oh, great, you've got my slides here, and it's exactly the right slide. You could do presentation mode if you like, Philip. And on this slide, we have on the left those are the 10% of the CpGs, some which are demethylated and some which are more highly methylated, left and the right, within a few hundred cases of childhood leukemia in our study. Forward the slide, please. Those changes are both increased methylation and demethylation at the promoter in some genes, and with the opposite profile in the gene bodies of those same genes. Next slide. These DNA methylation changes result in profiles that actually subclassify the leukemia in the same way that we subclassify them by cytogenetics. So hyperdiploid leukemias look similar to others, hyperdiploids, but different than ETV RUNX1 leukemias, for instance. Next slide. So first of all, let's look at the developmental origins. Are there epigenetic events that occur from exposures during pregnancy that then result in DNA methylation changes that are observable in the childhood cancer? Next slide. Now, why would we look at early environmental exposures during pregnancy? Well, this is why. There are waves of DNA methylation, demethylation, and remethylation during early development. On the left-hand side of this slide, we have the demethylation of the primordial germ cells, which occurs in the 3 months prior to pregnancy for men. In women, this occurs during her own fetal gestation, interestingly. Then there's a remethylation of those germ cells prior to fertilization. And on the right-hand side of this slide, those are the first few days after fertilization, showing that there's another wave of demethylation and remethylation. So really, by the end of the second week of pregnancy, we've got two of these waves of methylation, demethylation, which gives us a window. Next slide. Possibly a window to susceptibility to environmental factors, especially at this point very early in pregnancy. Next slide. Next, please. So our study where we perform our research is not a cohort study. It's the California Childhood Leukemia Study. This is a case control study. So cases were ascertained at the hospital, and controls were picked through the birth registry. Next slide. However, despite the fact that it's a case control study, we do have pre-diagnostic samples, and that's in the form of neonatal blood spots, which were taken at birth for all of the children that I'll show data on today. We call this the California birth cohort. The exposures that we've assessed in our study include dietary exposures, and we concentrate on the year prior to pregnancy. So in that way, we can capture, hopefully, the diet in those first few days of pregnancy where DNA methylation changes are so profound. We also capture pesticide exposures here, maternal drug exposures, and for a portion of our population, we have house dust and chemicals measured within the house dust. Next slide. The laboratory procedures are performing microarrays on 450,000 or 850,000, depending on which version of this array. Some of our data is based on the older version. These are a portion of CpGs around the genome. Not that many. There's about 23 million CpGs within the human genome. So we're just capturing a portion of it, but hopefully a portion that's illustrative of what's going on. Next slide. One thing we do, I won't talk about it too much more, but we need to understand that blood cells of different types of blood cells have their own distinct DNA methylation pattern. So whenever we look at environmental exposures, we need to adjust for those DNA methylation differences, and we do that using statistical methods to capture the DNA methylation profile of different blood cell types. Next slide. So first of all, talk about nutrients. And the nutrient that we're focusing on is mainly folate. Next slide. Now, folate has been assessed in animal studies, where obviously animal studies you can control exactly how much folate a rodent can consume during pregnancy, and then look at DNA methylation of the offspring. Those rodent studies have found that there are a few genes which are here in this on the bottom of the slide here. There's a few genes which are exquisitely sensitive to folate levels during pregnancy. And what this group here did in this is actually a human study, they took some of these genes and to see if they also varied in DNA methylation in response to folate during pregnancy. In an interesting study in the Gambia, where folate levels vary quite a bit by season, and you can see there's a dry season over here on the right, dry season and a rainy season. The dry season is when there's more folate available, and DNA methylation is generally lower in that dry season. Next slide. So we wanted to see if our California population was sensitive to folate that we assessed in a similar fashion. So we looked at the same genes and we found the same profile with lower DNA methylation at these folate sensitive CpG sites. So interestingly, kind of with the same profile as the Gambia study. Next slide. Now, why are we interested in folate and childhood leukemia? Well, in our California childhood leukemia study, we have seen that maternal diet quality score is related to leukemia risk. Here you can see a nice dose response. Next. And that's just to put in the arrows showing this dose response lowering of risk when we get to higher quality diets near the bottom, the fourth quartile, both for ALL and AML. Now, the Childhood Leukemia International Consortium has looked at folate. The one study that is sort of asked by almost nearly every childhood leukemia study is, did you consume supplemental folate or vitamin pills during pregnancy? And allowing us or allowing this group, Catherine Metier and colleagues, to do a meta-analysis of all the studies to show that there is a reduced risk with supplemental folate during pregnancy. Next. I just wanted to highlight this one group here. This is the parental education group, where those individuals that had none or primary school education seem to have the most profound effect of folate supplementation on leukemia risk. So the odds ratio there is 0.47 for that low education group. So we wanted to target that same group to see if we could also see that folate effect and see whether it may be mediated by DNA methylation. Next slide. Next, please. So just to illustrate that point, does DNA methylation mediate this effect? Next slide. So the CCLS, we have DNA methylation in our blood spots for over 1,000 cases, and did that Illumina DNA methylation array. Next slide. This is just an overall effect of folate in what we call differentially methylated regions around the genome. This is in particular with food folate. We quantify folate in different ways. We look at food includes what's in food and also what's in what's supplemented with flour products and corn products, for instance. And we see a number of sites around the genome. There's 30 here listed on the bottom, which seem to be associated or affected by folic acid intake. Next slide. The most profoundly affected site is DUSP22, and this is a gene known to be involved in leukemia. It's even involved in a particular translocation in one leukemia subtype. And the direction of DNA methylation is reduced DNA methylation in response to higher folate levels. And that's true for total folate and also for fortified folate. That's folate fortified in flour and wheat and corn products. And also, food-derived folate as well. Next slide. Now, again, as I mentioned, we wanted to target that group or take a closer look at the low income, also low education group to see what's going on with folate intake in that group and why it may be that supplementation is providing this or seems to provide this reduced risk of leukemia. Along the bottom here, we see profiles of dietary folate intake. Total dietary folate is on the left-hand side. Next to that, we have food-derived folate. And we also have the split up by ethnic group. So there's the Hispanic group and the non-Hispanic white group. Hispanics are the largest group within our California young population. So interestingly, Hispanics have higher folate intake levels in their food, in their diet. However, they have much lower frequency of taking prenatal vitamin or tablets exposure. That's over on the right here, supplemental dietary folate equivalents than non-Hispanic white subjects. Next slide. Interestingly, when they do take folic acid, they have more what appears to be a higher number of significant CPGs that are impacted by the folate intake levels. So we see three panels here. The one on the left is our total population. The one in the middle are the Hispanic subjects only, where 90 significant CPGs are indicated. And over on the right are the non-Hispanic white subjects. These are all the low-income subjects, so lower income, lower education, but we do see a higher impact of supplemental folate on this on the Hispanic group here, which kind of tells us this may be a group that we need to target for education or prevention with better dietary measures. Next slide. Now I'd like to talk a bit about another study that we've worked on. That's it's a study of twins. So in this study, we tried to capture twins in the California population by merging the cancer registry with the birth registry. And we wanted to see what was different about the twin who got leukemia compared to its identical match that did not get leukemia, with the hypothesis that it should be epigenetic differences since the twin pairs are genetically identical. Next slide. So you might say, well, the twins, they're genetically identical, they grow up in the same womb, they grow up in the same house. There shouldn't be any unshared factors, but there are. Nutrition is different even in monozygotic twins. The distribution of nutrition through the umbilical cord and placental function can be different. There can sometimes be different infectious exposures in one twin that are not that don't happen in the other. Next slide. Next slide. I'm going to speed up a little bit. So this is just how we diagnosed monozygosity, and it was using a SNP array, where on the left-hand side we see there's about half of our twins, 43 of them are genetically identical. The other 43 we did not include in this particular study of DNA methylation. Next. There were not significant differences with blood cell proportions between the twins in our studies. Thanks. I'm sorry, next. Next, please. Next. I just want to tell you, we did have a few genes which appeared to be reproducibly different between the twins, but on our next slide, I want to show what might be the most profound difference between the twins. Now, this is looking at overall DNA methylation in paired twins. The volcano plot on the bottom may look identical in the left hand and the right hand side of 0, but there is actually a bias towards the left. Push next again. And there is as we see on the right here, the negative coefficients outweigh the positive coefficients here. So by a binomial test, it's quite significant. So this may be telling us that there's some kind of subtle but profound DNA methylation difference in the twin who gets leukemia, that difference being lower DNA methylation. Now, where is that lower DNA methylation? The array we have has CpG sites in different regions of the genome, and some of them are in repetitive in the open C, repetitive elements in the open C. Some are in gene promoters or in the CpG islands or the gene body itself. Next slide. But what we found is that the biggest difference in our negative coefficients was in this open C repetitive regions in the backbone of the genome, indicating that there may be a reduced DNA methylation in the backbone of the genome, resulting in genetic instability and eventual leukemia in the one twin compared to its pair. Over on the left, you can see in the CpG islands that the bias is actually in the other direction. There's more DNA methylation in the promoter regions or the CpG islands in the twin who did not get leukemia. So the red here, the pink ones are the ones who did get leukemia compared to the greens who did not. Next slide. So again, what causes this epigenetic variation? It's probably some unshared factor in the monozygotic twins, such as nutrition. We really don't know yet, but we think it may be providing us some clues about leukemia genesis. Next slide. I'll keep pushing. Now what I've shown you first was DNA methylation related to disease. But what I'd like to show you next is an example of using DNA methylation to measure a dose of a particular chemical, which I think is really exciting for this group because so many of the studies have done such a careful examination of measuring chemicals. Next. Now, the easiest chemical to measure and assess for DNA methylation is tobacco. Next. And the reason is because this study, the PACE Consortium, which is the pediatric, it's a large consortium of cohorts that's looking at DNA methylation using a common array, and that's the same array that we're using. Next slide. And what this consortium found is that there are 3 particularly sensitive CpG sites to tobacco smoke in pregnancy that indicate smoking exposure that's affected the child. This DNA methylation in the newborn in cord bloods. Next slide. So just to make short story short, we developed an assay to look at these DNA methylation events in high throughput. So we looked at our genome-wide association study where we have 8,000 subjects genotyped and measured maternal smoking simply by looking at DNA methylation at these CpG sites. Next. Now, you might say, why would you look at smoking in childhood ALL? Because all of the studies are fairly negative. This is a meta-analysis showing this long history of negativity of maternal smoking in childhood ALL. However, in our own study, we are finding that there is a DNA methylation difference in at one of these CpG sites in the controls versus the cases, and the direction of that association is towards more smoking in the cases, and we are also seeing some subtype differences as well. Next slide. So now we're doing a genome-wide association interaction analysis with tobacco, and we're finding actually completely new SNPs or genetic polymorphisms that are completely unique from the usual ones that people have studied for ALL incidents in the past, that seem to interact with tobacco in potentially providing risk to childhood ALL. So it's really what we think is an exciting analysis and a good use of DNA methylation as a biomarker of an environmental exposure. Next slide. So just to review, I've shown two mechanisms or ways that DNA methylation can be used in these types of studies. One is to develop to study the developmental origin of disease with the hypothesis that an environmental exposure has caused an epigenetic switch that directly impacts the risk of the disease. And the second is simply to use DNA methylation as a biomarker of an exposure where the exposure may cause disease by other mechanisms besides epigenetics. Next. So in that way, it gives us a window to the past exposures. So we are studying a number of past exposures that are listed here. These are of particular interest in our population here in California. Yeah, again, I was really excited to see all the chemicals that people are examining in cohorts around the world. So I'm hoping we can actually come up with DNA methylation biomarkers for many of these exposures and then utilize them in our mechanistic studies in the future. Thank you.
So while I am queuing up, our final speaker is Dr. Yawei Zhang, who actually presented earlier in the cohort section, and I think is going to have some provocative and interesting ideas about investigating the causes of childhood cancer in China. So Dr. Zhang, I believe you can take it away if you're ready.
Thank you, Philip. Can you see my slides here?
Yes, I can.
Okay, great. Thank you, Philip, and thank you, Terry, for your invitation. I'm very glad and honored to present a strategy of investigating the causes of childhood cancer in China. As you all know, I was at Yale before, and I joined the China National Cancer Center last year, January, and formally become an employee here in China. So I'm continue collaborating with the China Beijing Children's Hospital, which is also the National Children's Health Center. So I'm presenting this strategy, and I also talked with the director, Xin Ni, and he's very happy to supporting this strategy. And we were hoping to implementing it, soon in China. As we all know, a lot of challenges in terms of investigating the causes of childhood cancer because of the rarity of the disease nature, and particularly for majority of childhood cancer, they are even rare. Currently, most studies are focusing on leukemia, but many other rare. I would say almost 90% of other childhood cancers has no opportunity to be investigated because of lack of power. And we all know the registry data, many studies have been relying on the registry data, but they do have limited exposure information. That's another major limitation. So for epidemiologists, we do know a cohort study will be ideal for study, the prospectively study causes, etiology of childhood cancer. And that's why we have this I4C being established. But we do know that for the individual study, cohort studies, the costs are really high, and also the potential for loss to follow-up. So now how we can, make better effort to establishing the cohort study. That's also been, a thing we have been thinking about it for many, many years. Finally, given the current, those new technology, the new medias and the internet, smartphone use, and a lot of app available. So make the job, it's possible. So over the next few minutes, I'm going to introduce a couple infrastructures we have been building in China. Mainly, we can make this, the infrastructure will be more attractive for people to participate in the cohort studies, and it would also make the entire cost, lower the cost, and also easy to follow up. So the first infrastructure I would introduce to you is we just finished this smart health management digital platform for primary cancer prevention. As you can see from this figure, this platform actually have 4 functions available. So the first function we built into this platform, and for individual users, they can answer a very simple questionnaire, and then they can evaluate their cancer risk, and also for other chronic diseases as well as pregnancy outcomes. For the pregnancy outcomes, we're still in the developing stage. Actually, we already finished, but I haven't really testing them yet. But for the cancer and other common chronic disease, this function already been done. And the second function, we develop the questionnaires. We make them as 17 different modules. So the users can use those, answering those questionnaires. They don't need to finish them at one time. They could do whenever they have a time. They could, working on those different modules. And also, we chopping, the kind of entire questionnaire to be a different smaller pieces for people will be able to finish them and make them feel a little bit easier. So the third function, we based on their information, they answered all those questions from those questionnaire. We can give them, delivering them the personalized prevention and intervention strategies. So the last function, we can, based on all the questionnaires, all the information they gave through the questionnaire, we can build in this large database as, our questionnaire data, so for the future use. And all those collections is not a one time. So we have a baseline, and we also have periodically sending the messages to follow up them. They can make this, updating their information. So this is the data manual we're going to be published in the Chinese Journal of Oncology very soon. So I just wanted to show you for the, the individual user, how that look like. It's, we build up under APP, and here is, we call the health management tab. So when people click on there, they will see their health evaluation tab. So they have a couple questions they can answer, then they can get their risk predicted. And also, this is all different modules for questionnaires. They can click any one to answer them. Just to show you a couple, this is Chinese version. This is a smoking, alcohol drinking, and diet. And this is their medical history, some family history, their reproductive history, and also physical activity, their residential history, etc. And also has an occupation as well. So this is just to show you when they answer the short questionnaire for their risk prediction. This is all the different cancers and chronic disease like cardiovascular disease and diabetes. For example, if they click lung cancer here, so they can get their risk. It's the below the general population, or it's greater than the general population. We also listed their protective factors for lung cancer and also their risk factors for predicting their risk here. So they can get some better sense, what make their risk high and what make their risk low. And also after they can answering each of those questionnaire modules, we give them individual feedback message. This is for alcohol. You can see we can evaluating their alcohol. They belong to the mild alcohol drinker or moderate or heavy drinkers. And then we also give them a message, suggesting them try to not drinking too much or even not drink at all. And also this is the feedback message for their evaluation for their hobby. And we evaluating their whether during their hobby they had exposure to heavy metals or some dust, and also for chemicals. And then we give them also suggested messages. For example, they can lower their frequency for hobby because those hobby has a potential harmful impact of this high exposure for chemicals. And also we suggesting them, wearing those protective stuff to, try to avoid, reduce their exposure levels. Just to kind of show you those two examples, you will get how they're using those, the individualized intervention strategies for them. So this is a major infrastructure we have built. We already finished this one, and we're going to launch a large cohort study in China. We'll starting in later March or early April using this whole platform. Another, for that infrastructure is mainly for the exposure sites component for collection. And another major infrastructure for China, they have built this National Center for Pediatric Cancer Surveillance, which started in 2019. And as we know, China had a tumor registry, but mainly focused on adult cancers. And for that system, which is running by the National Cancer Center here, so they only cover, I would say, less than 8% of childhood cancer patients. And we all know childhood cancer have a specific, the classification system, and they're mainly seen in the children's hospital and a specialized hospital. So for the adult cancer registry, they do not have those very tedious, or we would say very precise or high quality quality control the process for childhood cancer. So that's why China initiated this National Center for Pediatric Cancer Surveillance, which is affiliated with Beijing Children's Hospital. It's also the China National Center for Children's Health. And this surveillance system, started collecting the information. They started retrospective collecting starting from 2017. So that's they have data from 2017 until up to date. It's 2021, but still kind of keep ongoing to collecting the the new data. And this whole system it's about like 66% about all the pediatric cancer patients right now. And they're still expanding hopefully to capture, the all childhood cancers in China. And here just to show you, the example, this is their, you probably can see how their, patients migrating from one hospital to another hospital, seeking second opinion or seeking treatment approaches. So this is their, the registry pediatric cancer surveillance system. And also another network which is running by the Beijing Children's Hospital, and they established in 2016 called the Futang Research Center for Pediatrics in China, Pediatric Development. This network, they have 47 member hospitals, mainly the Children's Hospital, and some are maternity and a child care hospital. And all those member hospitals mainly came from the large or capital cities of different provinces. And also those hospitals, they had a network for lower level hospitals. Entire this system pretty much covering more than 80% of all pediatric patients in China. So all those infrastructures, they're building together will be really ideal for initiating China's national childhood cancer, the etiology cohort, consortium here. And we're using the first, we introduced those smart health management digital platform for primary cancer prevention. They can using this platform, for women to participating the system to answering all the questionnaires and get their exposure information quite detailed. And they could also using their Futan Research Center for Pediatric Development, this network to, recruiting women from the hospital. And also for the future, the outcome data, they can use in this National Center for Pediatric Cancer Surveillance to get the cancer outcomes. That will be, the whole preliminary strategy, I would say, investigating the causes of, childhood cancer in China. So this is the strategy, for China to, establishing the birth cohorts and the national birth cohorts to investigating causes of childhood cancer. I will stop here and just acknowledge a couple people who really make this whole strategy really, available in the near future. Particularly from the National Cancer Center, the director He Jie, for his great support, make my team here to realize this smart health management digital platform available. And also for Beijing Children's Hospital, the National Center for Children's Health and the National Center for Pediatric Cancer Surveillance, Director Xin Ni, and also the Deputy Director for Zhe Li, for their support and you share their registry data with me. And also my team who really make this whole digital platform available. Thank you for your attention.
I think we had terrific presentations and very stimulating messages. So I just want to thank you all, and that's it.
Thank you, Melina, and everyone else involved. Just really quickly, Philip, and thank you to you for taking on this chairing role. It's I know there are some uncertainties given that we're doing it in this novel way via Zoom. I know Zoom's not novel, but trying to bring together a global collaboration at a meeting is really something to manage, and the team at Turin have done a fantastic job. My closing comments would be that this has actually been incredibly motivating to me to see just how many people remain enthusiastic despite 2 years of lockdown, and to see the progress we're making, and also just to get the involvement of people who have had an involvement but not nearly as intense a one, and just a really exciting meeting for me. I think we really are making progress, and as we do come out of the lockdown, when we get back together, there will be things to do. One thing that occurs to me listening to Yahweh is that I've held this view for a long while, that one of the things we need to do with pediatric cancer is to actually measure the incidence well in a range of countries where, as you suggest, Joe and Yahweh agreed, we don't know, we really don't have very good incidence estimates from many of the countries that, like China, and China's better than a great many, that the cohort data can actually help with that. There's so many spin-offs from I4C that we didn't set out to achieve, but like that, large cohorts can give us reasonably accurate estimates of incidence. And even in China, what we're seeing there from Guangzhou, already with 30 cases, and it'll increase with their cohort of 50,000. We'll start to be able to better portray what's happening around the world, and that'll help us with our insights into causes and prevention. I don't want to keep people. For me, it's been a great a night and a day for me, actually, a great 2 days. So thank you, everybody.
Thank you very much.
Bye. Bye-bye.
Bye.